Cool. So week 3, guys, we're going to be going through wrist and hand anatomy and muscle physiology. So this is just kind of table of contents for it. So we'll go through bones first. So we'll start with your carpal bones. So they're your wrist bones. You have 8 of them and they're in 2 rows. So you have the proximal row, which is the scaphoid, then lunate. So they go across this way. So scaphoid, then lunate, triquetrum, and pisiform. And then the distal row is the trapezium, trapezoid, capitate, and hamate. So your hamate has a projection on the palmar surface, which is called the hook of hamate. And your pisiform is actually a sesamoid bone. So it's formed within the tendon of the flexor carpi ulnaris. So a mnemonic to remember it is straight line to pinky, here comes the thumb. And it's really good because it tells you which way it goes. So it goes around like anti-clockwise. And you will need to know all the names and where they are in the hand, but this will be easier once you do clin skills because they'll actually get you to like feel them on your hand. So it's easier to remember them then. Okay, then you have your metacarpal bones. So names are easy, just 1 to 5, 1 being the thumb. They have a head, the body, and the base. The sides, the medial and lateral sides of all the bones are concave so that the interossei muscles can attach. So they're just some of the muscles in your hand, which we'll go through in a bit. Then you've got your phalanges. So there's 14 of them. So these are your finger bones. So you have 3 in each of your fingers, but your thumb only has 2. So your thumb has the proximal and distal, and your other ones have proximal, middle, and distal. So these also make up 3 types of joints in your hands. So you have your MCP joints, which is your metacarpal phalangeal. So your metacarpals articulating with your proximal phalanges. You have your PIP, the proximal interphalangeal. So your proximal phalanges articulating with your middle phalanges. And then you have your DIP, which is your distal interphalangeal. So your middle phalanges articulating with your distal phalanges. So looking at these joints now, it will probably help more with what you learned last week with some of the forearm muscles. So like your FDP, your flexor digitorum profundus, that one does flexion at your DIP joints, but your FDS, your flexor digitorum superficialis, does flexion at your PIP joints. So that's a really big difference that you'll talk about more in clin skills. Okay, so that's just another diagram. Okay, joints. So the main joint is your wrist joint, which is also called the radiocarpal joint. So it's an ellipsoidal condyloid type synovial joint. So there's movement along 2 axes. So you have flexion and extension, and you also have ulnar deviation and radial deviation. You can also call it adduction, abduction, but ulnar and radial deviation is more common. So for the muscles that do all these actions, for your flexion, you've got both your flexors, well, all 3 of your flexors, so carpi ulnaris, carpi radialis, and digitorum superficialis. For your extension, you got your extensors, so they're kind of the opposite, like you got the carpi ulnaris and radialis for both. And then for your ulnar deviation, you've got both your ulnaris, carpi ulnaris, extensor, and flexor. And for your radial deviation, you've got both your carpi radialis, flexor and extensor, and also abductor pollicis longus. So the articulation in the wrist joint is the distal end of the radius over here, and this articular disc, which we mentioned last week. And they articulate with 3 of your wrist bones, so 3 of your carpal bones, the scaphoid, lunate, and triquetrum. So the articular disc, like we talked about last week, it just separates this distal radial ulnar joint from the wrist joint. So the ulnar doesn't actually articulate with any of the carpal bones. There are also 4 ligaments. So you have your palmar radiocarpal, dorsal radiocarpal, and your ulnar and radial collaterals. So the 2 radiocarpal ligaments, if you look at their names, they give a hint as to where they can be found. So the palmar radiocarpal comes from the palm side of your hand. It's on the palm side of your hand, from the radius to the carpal bones. And the dorsal is the opposite, on the dorsal side of your hand. And those 2 increase stability and ensure that your hand follows your forearm during supination or pronation. And your ulnar and radial collateral work together to prevent excessive lateral joint displacement. Okay, so your arterial supply is the branches of the dorsal and palmar carpal arches. So they basically just come from the ulnar and radial arteries. And your innervation is the anterior interosseous branch, which comes from the median nerve, the posterior interosseous branch from the radial nerve, and the deep and dorsal branches from the ulnar nerve. So yeah, you don't need to worry too much about remembering which individual branches, but there's some innervation from all three nerves. Okay, so your hand and finger joints. So we already talked about MCP, PIP, and DIP. The other one is the carpal metacarpal joints. So that's your carpal bones articulating with your metacarpal bones. And the movements that tend to happen at these joints include flexion, extension, abduction, adduction, and opposition. So just made these awesome diagrams which help show what the different movements are. Okay, so for muscles, there are 5 groups of muscles. The first one is thenar muscles. So these are 3 muscles at the base of your thumb, and they form this thing called the thenar eminence. So the 3 muscles are opponens pollicis, abductor pollicis brevis, and flexor pollicis brevis. So you can look at the names to figure out what they do. Pollicis just means thumb, and then opponens opposes the thumb, abductor abducts the thumb, and flexor flexes the thumb at the MCP joint. So the innervation for all of them is the median nerve, and the blood supply is the superficial palmar artery from the radial artery. So if you remember this, opponens, abductor, flexor. The next group is the hypothenar muscles. You'll notice it's the exact same. You have an opponens, abductor, and flexor. So these are the muscles that do your pinky. So they're located on the other side of your hand, and they form the hypothenar eminence. So digiti minimi just means your little finger. So you got the opponens digiti minimi, so that opposes the little finger. Abductor abducts, flexor flexes the MCP joint. So exact same as thenar, just pinky, not thumb. And your innervation is the ulnar nerve, and the blood supply is the deep palmar arch from the ulnar artery. So that's just a summary table. Okay, so lumbricals. So there are 4 lumbricals, 1 for each finger. They arise from both the medial or lateral surfaces of the metacarpal bones. And so to remember their action, I like to remember that they can make, that if you make a diamond with your hand, that's the action. So I don't know if you can see my camera, but like, if you do that, you're flexing at your MCP joints, but you're extending at your IP joints, so you can make like a diamond, just to remember the individual things. And then the innervation, so your median nerve does half of them, your lateral 2, and your ulnar nerve does the medial 2, so your ring and little fingers. So for your blood supply, it's the dorsal carpal arch and the superficial palmar arch. Now I've got your interossei. So there are 2 muscles located between the metacarpals. So you have your dorsal and your palmar interossei. So the dorsal one abducts your fingers at the MCP joint, and your palmar one adducts your fingers. So they also both assist the lumbrical, so the flexion at MCP and extension at IP. And then your innervation is the ulnar nerve. The blood supply for dorsal is the dorsal metacarpal arches, and the blood supply for palmar is the branches of the deep palmar arch. So these don't really fit in a category, they're just another couple of random muscles. So you have the palmaris brevis, which wrinkles the skin of your hypothenar eminence and deepens the curvature of the hand, which improves your grip. And you have the adductor pollicis. So if you look at the name, pollicis, thumb, adductor, well, adductor. So it adducts your thumb. And the innervation is the ulnar nerve. The blood supply for palmaris brevis is the superficial palmar arch, and adductor pollicis is the deep palmar arch. So that's just another summary table. And one thing you'll notice is for the interossei, the origins are different. So the dorsal interossei come from both the lateral and medial surfaces, while the palmar interossei come from the medial or the lateral surface. So if we go back a couple of slides, your dorsal ones, they come from both sides for the same muscle, but your palmar comes from either one or the other. Okay, so onto nerves. So your median nerve, which is C5 to T1, so that enters the hand via the carpal tunnel. So we'll go through the carpal tunnel more later on, but that's a very important area to know and very important fact. Okay, so the median nerve terminates by dividing into 2 branches. You have your recurrent branch and your palmar digital branch. The other branch that is in the hand is the palmar-cutaneous branch, but that comes off in the forearm and it doesn't travel through the carpal tunnel. So the motor functions. The recurrent branch does the thenar muscles, and the palmar digital branch does the lateral two lumbricals. So if you'll notice, the nerves in the hand are kind of the opposite to the nerves in the forearm. So in the forearm, for the anterior compartment, we had ulnar doing only flexor carpi ulnaris and half of another muscle, but median did everything else. So median was the main one. In the hand, it's the opposite. Your ulnar nerve is the main one, and your median just does the thenar muscles and the lateral two lumbricals. So that basically just flexes the digits. So for sensory functions, your palmar-cutaneous branch does this blue area, and your digital cutaneous branch does this green area. So in terms of sensory innervation of the hand, it's really important to know which parts radial, median, or ulnar, but the individual branches aren't as important. It's more important to know between the main three. So your ulnar nerve, it travels superficial to the flexor retinaculum and medial to the ulnar artery. So it enters the hand via this ulnar canal, and it terminates by giving rise to superficial and deep branches. So the motor functions, the deep branches do basically all your hand muscles except for the thenar and half your lumbricals. So everything else, your hypothenar, two lumbricals, interossei, and your adductor pollicis and palmaris brevis. For sensory innervation, the ulnar nerve, the palmar branch does this area here, yellow area, the dorsal cutaneous does the green area, and the superficial does the blue area. Basically, remember your ulnar bone is on the pinky side of your forearm, and so it does basically both sides of the palm and fingers on the front and back of your hand on that side, so 1.5 fingers. And so your radial nerve only has sensory functions in your hand. So it does this red area. So this just shows them all together. So your ulnar is on your pinky side, so you got the 1.5 on front and back. Your median does most of the rest of the 3.5. So it does on the front, and it does the fingertips on your back, and the radial does most of it on the back and just a little bit on the side at the front. So you can remember that by thinking radial usually does the posterior aspect all along your upper limb. Like for forearm, it innervates the posterior part as well. So it continues that into the hand. And median in the forearm did anterior. So here it's sticking with anterior. So your hand has a rich arterial supply. So this allows your hand to be supplied even when under high resistance to flow. So like when you're grasping something or applying pressure. So you have your two main arteries, so your ulnar and your radial artery. And your ulnar artery enters your hand anteriorly to the flexor retinaculum and laterally to the ulnar nerve. So it has a deep branch that comes off it, while it actually continues as the superficial palmar arch. The radial artery, it enters your hand dorsally. It crosses the floor of the anatomical snuff box, which is an area we'll go through later on. And then it turns medially and travels between the heads of the adductor pollicis muscle. So that has a branch to the thumb index finger, and it also has a superficial palmar arch, while that continues as the deep palmar arch. So you notice both the ulnar and radial arteries, they both have a branch to the other arch, but the ulnar continues as superficial and the radial continues as deep. So your superficial palmar arch is anterior to the flexor tendons and deep to the palmar aponeurosis. So it has digital arteries which come off it, which supply your 4 fingers. And then your deep palmar arch is deep to the flexor tendons, and it helps supply your digits and wrist joint. For venous drainage, you've got pretty much the opposite of arterial. So you've got a superficial and a deep venous palmar arch. And so in the dorsum of your hand, you can see over here, you have your dorsal digital vein, which drains into your 3 dorsal metacarpal veins, and these veins unite to form a dorsal venous network. And this network, you get the cephalic and basilic veins arising from them. And these ones are the ones that continue into your forearm and arm. So most of the blood in your hand goes through this network into the cephalic and basilic veins. Okay, so you have 3 anatomical areas in your hand that are of importance. The first one is the carpal tunnel. This one is very important. There's a very important clinical pathology when you get carpal tunnel syndrome associated with it, and they like to test you on that a lot. So the carpal tunnel is basically a passageway in the anterior portion of your wrist for your tendons and your median nerve. So the borders of it are the deep carpal arch, which is a concave surface. So that's just your carpal bones. So you have the scaphoid and trapezium tubercles and the hook of hamate and pisiform. And then the superficial flexor retinaculum. So that's just connective tissue, and it basically just goes across the top to convert the arch into a tunnel. And then you've got the contents going through. So the contents, there are 9 tendons and 1 nerve. So your median nerve, and then the 9 tendons, you have your tendon of flexor pollicis longus. Then you have your 4 tendons of FDP and 4 tendons of FDS. So one way you can remember it is that because the arch carpal tunnel is on the anterior surface of your hand, that's why you have the that's why you have the flexors going through. So there are 2 synovial sheaths which allow free movement of the tendons. So the FPL tendon has its own sheath, while all 8 of the FDP and FDS tendons are in another sheath. Okay, for your extensor tendon compartments. So this is on the back of your hand, and there's basically 6 tunnels which transmit the extensor tendons into your forearm. So they're not as important, so you don't really need to remember what's in each one. But the more important part is that the compartment 1, which contains your extensor pollicis brevis and abductor pollicis longus, is the lateral border of your anatomical snuff box. Compartment 3, which is the extensor pollicis longus, that's the medial border of the anatomical snuff box. And then compartment 2 and 3 are separated by Lister's tubercle. Otherwise, it's not as important to remember what's in each compartment. I don't think they ever really tested us on that. Okay, yeah, anatomical snuff box. So that's just a triangular depression on the back of your hand. So if you extend your thumb, you'll be able to see just a triangle just above your wrist. And so the borders, like we just went through, the medial is the extensor pollicis longus tendon. The lateral is the abductor pollicis longus and extensor pollicis brevis tendons. The tendon of, yep. And then the proximal border is the styloid process of the radius. And the floor is your carpal bones, so your scaphoid and trapezium. So they're your 2 rightmost carpal, they're your 2 carpal bones at the very end of the line. And then the roof is the skin. So the contents, the radial artery crosses the floor, then it turns medially to go through the heads of the adductor pollicis muscle. And then you have your superficial branch of the radial nerve, which is actually in the skin and the subcutaneous tissue. Then your cephalic vein actually comes out of there to travel up your forearm. Okay, so that's it for anatomy. I'll be going through physiology, so movement and motor control now, and then there will be more questions at the end. Before we do that, does anybody have any questions for anatomy? Nope, okay, cool. Okay, so a bit of muscle recap. So the properties of the muscle cells are basically generate force and to shorten. And the functions, they produce body movements, maintain posture and body position, and stabilize joints and generate heat. So characteristics, excitability, so the ability to receive and respond to a stimulus. Contractility, ability to shorten forcibly when stimulated. Extensibility, so to be stretched when relaxed. And elasticity, so the ability to recoil to resting length after it has been stretched. So for your types of muscles, 3 types from last time that you should know. So skeletal, cardiac, and smooth. So your skeletal and cardiac muscles have striations, but your skeletal has ordered striations, so your contractions occur in one direction, but your cardiac has branching and unordered striations. So your contraction occurs on multiple axes, and your smooth just doesn't have any. So your skeletal muscle is under voluntary control, while the other 2 are involuntary. In terms of nuclei, the skeletal muscle has multiple nuclei on the periphery of the cells, while the cardiac muscle has a single central nucleus per fiber. And your smooth muscle has a single nucleus per fiber. Okay, so a closer look at skeletal muscle. So you have one skeletal muscle, which has a bundle of muscle fibers inside it, which splits up into myofibrils, which has many sarcomere units. And your sarcomere units contain your actin and myosin myofilaments. So hopefully you remember this diagram from last semester. So this is a sarcomere, and the Z lines mark the boundaries of the sarcomere, while the M line is the middle. The A band is all of myosin, so including the overlap with actin. It's the total length of myosin. Your I band is just the actin part. So when your muscle contracts, in most cases, the I band is the part that shrinks. And then your H zone is only myosin area. Okay, so onto control for movement. So we have excitation, contraction, coupling. So excitation is the stimulation by the neuron, and contraction is the interaction between actin and myosin. And then there's relaxation. So it starts in the NMJ, the neuromuscular junction. So what happens is your action potential travels down the motor neuron, and then the ACH is released, and it binds to the receptors on the motor end plate, which is basically on the target skeletal muscle. And then there's an influx. This causes an influx of sodium at the motor end plate. So the action potential travels along the plasma membrane, and it goes down the T tubules. This is where it hits the voltage-gated DHP or dihydropyridine receptors, and they open, and this opens the ryanodine receptor. So the DHP and ryanodine receptors are physically coupled Ca2+ channels. So this causes the sarcoplasmic reticulum to release calcium. So at rest, so we'll go through cross-bridge cycling now. So at rest, actin is blocked by tropomyosin and troponin, while myosin is unbound, but it's energized. So the first step of cross-bridge cycling is that calcium released from the sarcoplasmic reticulum binds to the troponin. So this causes the tropomyosin to be pulled away, and this exposes the myosin binding site on the actin. So the myosin binds to the actin, forming a cross-bridge. And then in the power stroke, the myosin head rotates, releasing ADP and PI, as you can see over here, and then the actin strand is pulled, so the sarcomere contracts. And then ADP binds to the myosin, which breaks the cross bridge, which is the link between the actin and myosin. And then the bound ATP is split, re-energizing the myosin head. So that's just another diagram. Okay, and then for relaxation, calcium ATPase pump causes uptake of the calcium back into the sarcoplasmic reticulum. So this goes against the concentration gradient. And without any calcium binding to troponin, tropomyosin falls back into place and it covers up the binding site on actin. So you can't form any more cross bridges. Okay, so this is just a summary. So the main things to note are the action potential in the motor neuron starts everything. And then the calcium binds to troponin, which starts that whole sequence of contraction. And then the ATP binds to myosin, which starts breaking the cross bridges. So your sliding filament theory. So this basically says that the muscle contraction caused by actin is caused by actin and myosin sliding against each other. So yeah, basically it's just them sliding and there's no change in the actual length of each filament. And the sarcomeres always pull towards the center. So length tension relationship. This is active tension. So there's an optimal length where the overlap between actin and myosin is basically optimal. So this occurs in resting state, and this is the best position for tension building. If you look at the maximum length, so there's no overlap at all between actin and myosin, then the amount of tension you can build is less. And if you look at the minimum length, so there's maximum overlap between actin and myosin, and even a little bit of overlap of the actin on either side, then that's also decreases the amount of tension you can produce. So we also have this thing called passive tension, which is your Titin filaments. So these Titin filaments, they're these yellow springy bits down here. So they help give muscle tension and elasticity and help us return to the relaxation state. Okay, so just a quick note on rigor mortis. So that's stiffness after death. So how this actually occurs is that there's no ATP available, so the cross bridges are locked in the contracted position. Because if you remember, the ATP binding to the myosin is what broke the cross bridge. And because of that, the muscle stays stiff for a few hours. Eventually, the muscle fibers degrade, so the stiffness goes, but the length of rigor mortis depends on the situation. So for example, if you're exercising, then you've used up a lot of ATP, so it lasts for longer. So we can classify contractions into isotonic and isometric. So if you look at the name, you can tell which type is which. So iso just means same, and then tonic, tension, metric, length. So isotonic, same tension, but the muscle changes length. So for example, picking up a heavy box or doing bicep curls. For isometric, same length. So you can develop tension without changing length. For example, pushing down on a table or doing wall sits. So basically equal and opposite forces develop tension. At a molecular level, this means that cross-bridge cycling always occurs when muscle, so basically cross-bridge cycling always happens whenever a muscle is stimulated, whether it's lengthening or there's tension, developing tension, it's always happening. If there's no lengthening, the cross bridges just rebind in the same place. So your isotonic contractions can be further split up into concentric and eccentric. So the concentric contractions is when the muscle is shortening while contracting, like lifting up a water bottle, and eccentric is when the muscle is lengthening, so putting down a box. So we'll go through a bicep curl as an example. So before you start movement, if you're just holding the weight in your hand, that would be an isometric contraction because there's no movement, so your muscle length isn't changing, but you're holding the weight, so you're developing tension. When you actually do the movement of the bicep curl, then it's overall isotonic because your muscle length is changing, but the tension isn't. So the up part, as you can see here, that would be concentric. So your muscle is shortening. And the down part would be eccentric because your muscle is lengthening. Okay, so motor control. So we have neurons and cell bodies in the ventral horn for motor control, and then the size of the ventral horn depends on the level of the spinal cord. So where you need more complex control, like in your hands, there will be a larger ventral horn at that level. So the cell bodies that innervate the proximal muscles are closer to the midline, and the cell bodies that innervate the distal muscles are further away. So your alpha motor neuron. It has a lot of different names, so it can also be called a lower motor neuron, a motor neuron, or a final common pathway. That's basically this last neuron that comes from the ventral horn down to the skeletal muscle cell. So this is involved in all movement, reflexive and voluntary. So it directly innervates the muscle, and yeah, the cell bodies are in the ventral horn. So your motor unit is one alpha motor neuron. So like you can see here, this purple one, it's one alpha motor neuron, and the muscle fibers it innervates. So all these purple muscle fibers and that neuron would consist of one motor unit. So you can have small ones, which are less than 10 muscle fibers. So for high control, like in your fingers or eyes. But you can have large ones, which are like over 1,000 muscle fibers, which is a lot. And those would be for power, like in your calf muscles. You can also have, there's also this thing called a motor neuron pool. So that's just doing the opposite. You take one muscle and the collection of alpha motor neurons that innervate it. So there's two ways to control force generation. You have rate coding and you have the size principle. So rate coding is basically controlling the frequency of stimulation. So you're alternating the time between each stimulus to control contraction. So it's measured in hertz, which is basically the number of cycles per second. So the higher the number of hertz, the more cycles. So less time between each stimulation. So we'll start over here with 5 hertz. So that means it's called a twitch force because there's enough time for the muscle to relax before it's stimulated again. If we ramp it up a bit, so 10 hertz, then it's temporal summation. So the action potentials are building on each other. So it comes up like that. So before the muscle can fully relax, it's been stimulated again. If you go up a bit more, 25 hertz, it's unfused tetanus. So that's when you reach the certain level where there's just almost no time for the muscle to relax in between stimulations. And go up even further, it's fused tetanus. So this one's oscillations, but this one's just smooth. So for your size principle, so different motor units have different number of fibers. So some of them are small, might only have 3 fibers. Some of them are large, so they have 7. So what happens is you recruit your small ones first, and then if you need more force, then you can recruit larger ones. So the smaller ones are fatigue resistant because they're the ones that are recruited first. And they have a greater firing rate, for example, up to 24 hertz. The larger fibers are not fatigue resistant. So those have a lower firing rate, for example, up to 16 hertz. So you'd start with getting like these X fibers first. And then if you need more force than that, you'd start getting Y. And then if you need even more, then you'd start getting the Z ones. So in general, to increase force, 2 things to do. You can increase the frequency of stimulation, and you can recruit more and larger muscle fibers. So to look at the difference, to pick up an egg, you don't need much force at all. So you don't need to fire often, and you can use only your small motor units. But if you want to crush an egg, that requires a lot more force. So it's a higher firing rate, and it uses both smaller and larger motor units. Okay, question time. So just write your answers in the chat. So does anybody have any idea what the actions of the radiocarpal joint are? Okay, so for a clue, your radiocarpal joint is, so if you look at the name, radius carpal bones, so it's your wrist joint. So, I mean, just try moving your wrist around, see what you can do. Yep, that's good. So, yeah, flexion, extension, and then abduction or adduction. Yeah, so using those terms are fine, but yeah, if you ulnar deviation and radial deviation are more common to see, but they're both correct, so it doesn't matter which you use. Okay, so what are the actions of lumbricals? Yep. So you can remember the diamond shape, just flexion and then extension. Good job. Okay, so which nerves innervate which area? Yep. So good job. Remember which side things are. So your ulnar nerve, pinky side, so that has to be green. And then radial nerve is posterior, so on the thumb side, so red, and median's got the weird just fingertips on the back and your anterior side on the thumb. So that's blue. So contents of the carpal tunnel. Yep, B is good. So yeah, that's just really important, something you'll have to remember. Also, the palmaris longus doesn't go through the carpal tunnel, and flexor carpi ulnaris tendon doesn't go through the carpal tunnel either. Okay, so what forms the floor of the anatomical snuff box? Yep. It's C. So straight line to thinking, here comes the thumb. So that's the mnemonic you can use to remember. And then, yeah, it's just the first and last. So the straight and the thumb. So it's scaphoid and trapezium. Okay, so muscle physiology. So which of the following is false? So the I band is only actin, so excluding the overlap. Good job. So what's the first step of cross-bridge cycling? Yep. Nice. So yeah, calcium binds to troponin, which starts the whole sequence of the tropomyosin moving, and then the myosin binding to actin and everything. So if you put down a water bottle, what type of contraction is it? Yep, good job. So if you look at the names, you remember isotonic, same tension, changing length. Isometric, same length, changing tension. Eccentric and concentric are whether it's the muscle is shortening or lengthening. And so considering the muscle is changing length, they both have to be isotonic. So you can't have isometric and eccentric. That's like contradicting itself, like same length and also changing length. So just remember they come from isotonic. And then yeah, muscles lengthening while contracting if you're pulling down a water bottle with the same tension. So what type of force is this? Yep. Infused tetanus. Okay, great. That's all the questions for today. So next week we'll go through some clinical overview for upper limb and some sensory and neural integration, plus the MSK exam for clinical skills. Thanks for coming, guys. Stay behind if you got any questions.